Carbohydrates are a fascinating class of biological molecules and a critical part of human diet and nutrition. Carbohydrates can be easily recognized by the multiple hydroxyl groups attached to their carbon frame. In fact, carbohydrates have as many oxygen atoms as they do carbon atoms, and twice as many hydrogens, giving them the common formula of CH2O. From this formula, it is easy to see where carbohydrates get their name, which literally means watered carbon. As you might guess, all those oxygens make carbohydrates very soluble in water. Their primary purpose is storing energy, but some important structural molecules in the cell are also made from carbohydrates. Carbohydrates can be divided into four major categories: monosaccharides, disaccharides, polysaccharides, and oligosaccharides. The suffix saccharide comes from the Greek word for sweet. We will present and review the vocabulary, properties, and principles related to each of these important carbohydrate groups. Monosaccharides are the most basic carbohydrates. The following section will discuss the structure and chemical groups of monosaccharides, as well as their nomenclature. We'll also demonstrate how monosaccharides can switch between a ring formation and a linear formation. Finally, we'll discuss some of the common monosaccharides that you need to know as you go through biology. Monosaccharides are also known as simple sugars. They function primarily in the body as an energy source, but they can also serve as building blocks to make larger carbohydrates such as disaccharides, polysaccharides, and oligosaccharides. The monosaccharides can be characterized by their many hydroxyl groups. Each monosaccharide will also have a carbonyl group on it, shown here in red. Sometimes that carbonyl group can be difficult to identify, particularly when it's in the ring structure. On this slide you can see several common names: glucose, galactose, fructose, and ribose. Notice that each of these end in the same OSE suffix. The OSE suffix is reserved for the simple sugars, the monosaccharides, and the disaccharides. Monosaccharides come in many different types that differ in the number of carbons they carry and in the location of their carbonyl group. Monosaccharides that carry 7 carbons are called septoses. If it has 6 carbons, it's called a hexose. 5 carbons are called pentoses. Tetroses are 4 carbons, and trioses have 3 carbons. We most often see monosaccharides having either 6, 5, or 3 carbons. Monosaccharides are also named by the location of their carbonyl group. If the carbonyl group is on the end of the carbon chain, the sugar is called an aldose sugar, which is a derivation of the term aldehyde you learned in a previous lesson. Sugars with the carbonyl group inside the carbon chain are called ketoses. Both the number of carbons and the location of the carbonyl group can be described together in a single word, like hexaketose or penta-aldose. Monosaccharides can be drawn in 2 different forms. The linear form, seen here on the left, makes it easy to identify the carbonyl group and the arrangement of hydroxyl groups. However, in solution, the partial negative charge on the 5-hydroxyl group is attracted to and attacks the partial positive charge on the carbonyl carbon. When this happens, a new covalent bond forms and the molecule rearranges into a ring structure. The ring is composed mostly of carbons, but there will always be an oxygen atom that is part of the ring itself. This is characteristic of carbohydrates. In nature, the molecule can fluctuate between the linear and ring structures, but the ring structure is chemically more stable, and the monosaccharide will be in the ring form about 99% of the time. As a result, you will most often see monosaccharides drawn as a ring structure. The ring structure can sometimes be hard to read because all those carbons, hydrogens, and hydroxyl groups seem so packed together. To make things less busy, you often see monosaccharides in their abbreviated structure, in which the hydrogens and the carbons have been removed. In this representation, the intersection of two lines indicates a carbon atom. Any missing bonds on carbons are assumed to be occupied by hydrogen atoms. Here's the abbreviated structure for one of the most common monosaccharides, glucose. Glucose is the first reactant in glycolysis and one of the components of table sugar. Glucose is an aldehexose. It has 6 carbons, and the carbonyl group in the linear form is located at the end of the chain. Galactose and mannose are also common aldohexoses. Both are important building blocks for disaccharides and oligosaccharides that we will learn about later. What's interesting about galactose and mannose is that their structure is so similar to glucose. Still, there are differences. Take a moment and see if you can spot them. If you said that this hydroxyl group on glucose and galactose is pointing in different directions, you're right. Mannose also has a unique hydroxyl group, but it is located on a different position on the ring. This makes both galactose and mannose structural isomers of glucose. Some other notable monosaccharides are fructose and ribose. Both have a pentameric ring, but fructose is a hexose, while ribose is a pentose. Fructose is another important intermediate in glycolysis, as well as a building block for the disaccharide sucrose. Ribose is a building block for RNA found in the cells. If you remove the number 2 hydroxyl group from ribose, you get deoxyribose, which is an important building block in making DNA. Now, let's review the things we have learned about monosaccharides. Monosaccharides are the simplest carbohydrates and serve as a building block for larger molecules. They are made of the common molecular formula CH2O. Monosaccharides always have several hydroxyl groups on them and one carbonyl group. Sometimes the carbonyl group in the ring structure can be difficult to find. All of the oxygens present in those groups make monosaccharides very soluble in water. Monosaccharides are named by the number of carbons. Tri, penta, and hexa are prefixes used to denote carbon chains with 3, 5, and 6 carbons respectively. Monosaccharides are also identified by the location of a carbonyl group. Aldoses are monosaccharides that contain the carbonyl group at the end of the chain, while ketoses are monosaccharides that contain the carbonyl group somewhere in the middle of the chain. Monosaccharides can convert between a linear and a ring form. The ring form is more stable, and in nature, the monosaccharide is about 99% in the ring form. Abbreviated structures are often used to more easily view monosaccharide rings. These structures lack the carbons and hydrogens within the structure. There are several common monosaccharides, but the most common is glucose. It is used in energy production as an important building block in polysaccharides that we'll learn about later. Galactose and mannose are structural isomers of glucose. They are important building blocks in their own right for several disaccharides and oligosaccharides. Ribose, deoxyribose, and fructose are unique in that they all form pentameric or 5-sided rings. Ribose and deoxyribose are important components of the nucleic acids DNA and RNA. Fructose is an important monosaccharide in its own right and is used heavily in energy production and as a building block for disaccharides. This completes our study of monosaccharides. You can now move on to the next section about disaccharides. A disaccharide is a simple sugar created from a condensation reaction between two monosaccharide subunits. In this tutorial, we will describe that condensation reaction, which results in a bond called a glycosidic linkage. An important element in glycosidic linkages is the anomeric carbon. We will define anomeric carbons, learn how to identify them, and discuss their impact on carbohydrate chemistry. Glycosidic linkages are named according to their participating carbon numbers. You will learn how to number carbohydrate carbons and then name the resulting glycosidic linkages. Finally, we will introduce you to several common disaccharide examples. A disaccharide is formed when two hydroxyl groups from different monosaccharides are combined in a condensation reaction. Water is released and the resulting bond is called a glycosidic linkage. Glyco is a Greek root for sweet. Each glycosidic linkage always involves a special carbon in the chain identified as the anomeric carbon. The anomeric carbon is special because it is the location of ring forming and breaking. It is also essential in forming glycosidic linkages. This means that at least one of the carbons in a glycosidic linkage must always be an anomeric carbon. The simplest way to identify the anomeric carbon in a ring structure is to look for the carbon that is bound to two different oxygens. One of those oxygens is almost always the oxygen found in the ring itself. The second oxygen will be in a hydroxyl group or a glycosidic linkage. The anomeric carbon really only exists in the ring formation. In the linear structure, it becomes the carbonyl carbon. Identifying anomeric carbons will be a useful skill in understanding future lessons. Practice identifying the anomeric carbon in the monosaccharide below. You will be given 10 seconds to complete the activity. If you need more time, simply push the pause button. Hopefully, you were able to identify the highlighted atom as the anomeric carbon with two oxygen bonds. Now try this one. Here is the proper anomeric carbon, and here are the two oxygen bonds that identify it. Here's a final practice. The orientation of this one makes it a little harder. The anomeric carbon is here, and the two oxygen bonds are here. Notice the anomeric carbon in all three examples was attached to the ring oxygen. Polysaccharides are polymers of monosaccharides that can stretch for hundreds of subunits. As with disaccharides, they are formed from sequential condensation reactions. In this tutorial, we will discuss two different families of polysaccharides: the storage polysaccharides like glycogen and starch, and structural polysaccharides like cellulose and chitin. Important features will include branching and the alpha-beta stereochemistry introduced in the disaccharide tutorial. Storage polysaccharides, like the glycogen molecule shown here, are used for storage of energy. The monosaccharides in a storage polysaccharide are chemically stable for long periods of time, but they can be broken off by enzymes and rapidly degraded to meet the cell's need for ATP. In contrast, the second group, called structural polysaccharides, are not broken down very well by the body. They tend to form tough, fibrous strands that are used to strengthen cellular structures. Cellulose in plants is just one example of a structural polysaccharide. Storage polysaccharides are more commonly known among the general public as complex carbohydrates. Made exclusively from glucose monomers, storage polysaccharides can be made from hundreds to thousands of glucose molecules. In all cases, the glucose monomers are held together primarily by alpha-1,4 glycosidic linkages. The simplest storage polysaccharide is amylose, a type of starch found in plants. The bond angles of the alpha glycosidic linkage cause the glucose monomers in amylose to form a helical structure that ends up looking like a telephone cord. Amylopectin is another starch very similar to amylose, with the exception of occasional polysaccharide branches that form off of the main chain. Branching occurs when a glucose monomer forms an alpha-1,6 glycosidic linkage in addition to the 1,4 linkages that make up the main chain. The 1,6 linkage creates a branching point for a new chain. Note that these 1,6 linkages are important as branching points, but they occur rarely in comparison with the 1,4 linkages that make up the chains themselves. Amylose and amylopectin are both starches, and they are only found naturally in plants. The primary storage polysaccharide in animals is called glycogen. It has the same alpha-1,4 linkages and structure as amylopectin, but has even more branching. Having lots of branches creates multiple free ends for enzymes to release glucose for energy production in a hurry. But all that branching makes it difficult for glycogen molecules to efficiently pack together. In contrast, plants like this potato rarely need large bursts of glucose-driven energy. Instead, they need a way to store lots of glucose in a compact space. The unbranched structure of amylose is a great solution because with limited or absent branching, it can pack a lot of molecules together, even if they are not so easy to access for energy. To illustrate, this image shows glycogen and amylose together. Both had the same number of glucose subunits, but the amylose can pack them in less than half the space needed for glycogen. Whether branched or unbranched, a defining feature of the storage polysaccharides is the alpha linkages that hold them together. This is significant because animals like horses, hummingbirds, and hippos have enzymes capable of breaking these alpha linkages to create the glucose needed by the cell for ATP production. In contrast, the structural polysaccharides are made with beta-1,4 linkages. Multicellular animals lack an enzyme capable of breaking beta linkages, making the resulting fibers difficult to break down for energy, but perfect for building tough, fibrous structural elements in cells and tissue. One of the most common structural polysaccharides is cellulose, which gives grass and trees their tough, fibrous characteristics. If animals can't break down structural polysaccharides like cellulose, you might wonder how animals like cows or termites are able to live on grass or wood. Such animals actually carry special microorganisms that live in their gut and excrete the necessary enzymes to break beta linkages and release the energy-producing glucose for consumption. The structural polysaccharides are commonly known as dietary fiber. They are characterized by beta-1,4 linkages. As previously discussed, this type of bond makes the resulting polymers indigestible to most animals. The bond angles of the beta-glycosidic linkage also creates a different polymer structure than the helical coil seen in the storage polysaccharides. Beta linkages create long, uncoiled strands. This feature partially occurs because the beta bond angles require that every other sugar in the chain is flipped upside down. Note the orientation of the ring oxygen changes from one monomer to the next. Several strands are capable of hydrogen bonding with each other through the multiple hydroxyl groups on the monosaccharide subunits. The resulting fibers become very tough and difficult to break or pull apart, making them ideal for building durable cell structures. Cellulose is the most common structural polysaccharide in plants. Like starches and glycogen, it is made exclusively from glucose monomers. It's amazing that two molecules made from the exact same monomer can produce such different structures just by changing the orientation of the anomeric carbon from alpha to beta. Chitin is another common structural polysaccharide. Chitin is a key component of the hard exoskeleton of most arthropods, including crabs, lobsters, and beetles. Chitin is made from N-acetylglucosamine monomers. This is a modified version of glucose where an amine group replaces one of the hydroxyl groups. You might wonder if there are any structural polysaccharides in animals. The answer is yes. Structural polysaccharides, called glycosaminoglycans, are important components of the extracellular matrix of different tissues. One common glycosaminoglycan is hyaluronate, which has a repeating disaccharide unit composed of glucouronate and N-acetylglucosamine. As you might expect, the subunits are held together by beta linkages, making them long-lasting structural fibers. Now, let's review what we have learned about polysaccharides. Polysaccharides are long polymers of monosaccharides with hundreds to thousands of subunits. Storage polysaccharides are one family of polysaccharides that includes starches and glycogen. They all share a common alpha 1,4 linkage type that causes them to form helical structures. 1,6 alpha linkages in storage polysaccharides leads to branching. Glycogen is highly branched, which is better for energy release, but not for efficient storage. Structural polysaccharides like cellulose, chitin, and glycosaminoglycans are made from beta linkages. Beta linkages form linear chains that hydrogen bond to each other to form strong fibers. Animals can digest alpha linkages in storage polysaccharides, but most lack the enzyme to digest the beta linkages found in structural polysaccharides. Oligosaccharides are the fourth and final group of carbohydrates included in this series. Oligosaccharides are smaller and more diverse polymers than their polysaccharide cousins. This tutorial will discuss their basic properties as well as their location and function in the cell. Oligosaccharides are a unique group of carbohydrates. Although they are polymers of monosaccharide subunits, they are much smaller than their polysaccharide cousins and usually range from 3 to 20 subunits in size. They are also much more diverse in their monosaccharide composition. Here are just a few of the more common subunits. Each color represents a different monosaccharide. Notice from these examples that the oligosaccharides also typically have more branching. Oligosaccharides are associated with cell membranes and are always attached to a protein or lipid at the membrane surface. The resulting structure is called a glycoprotein or a glycolipid. These examples are not drawn to scale. The oligosaccharides would be much smaller, but they do illustrate the extensive diversity and branching of oligosaccharides. Another unique characteristic of oligosaccharides is their cellular location. Oligosaccharides are always found on the extracellular surface of the cell or the luminal surface of organelles. They are never found on the cytosolic surface of either. The diversity of subunits in oligosaccharides makes them good markers for cell identification, and this is one of their primary purposes. The oligosaccharide pictured here is a glycolipid made of 6 different monosaccharides. It is found on the surface of red blood cells and determines the ABO blood type of an individual depending on the identity of the last subunit highlighted in yellow. Those with the N-acetylgalactosamine subunit in the terminal position have type A blood. If the N-acetylgalactosamine is replaced with a galactose, it produces type B blood. If neither is present, then type O blood results, and if glycolipids with both markers are found, then type AB blood results. If an individual is given blood with a foreign glycolipid, the immune system easily identifies those cells as foreign and immediately initiates a strong immune response that in most cases leads to severe shock and even death. Oligosaccharides can also act as a form of protection. For example, oligosaccharides on the cell surface can keep harmful bacteria or viruses from being able to land on the cell and infect it. Oligosaccharides can also help cells protect themselves on the inside. Cell digestion takes place in an organelle called the lysosome. The enzymes responsible for digestion are kept from digesting the lysosome walls and each other by having lots of oligosaccharides attached to the inner surface. Now, let's review what we've learned about oligosaccharides. Oligosaccharides are short carbohydrate polymers from 3 to 20 monosaccharides that are covalently attached to proteins and phospholipids. Oligosaccharides are characterized by a diverse array of monosaccharide subunits and by extensive branching. Oligosaccharides are found on the extracellular surface of the plasma membrane or the lumen of organelles. The ABO blood types result from a variation in a red blood cell glycolipid. Type A blood contains an N-acetylgalactosamine terminal residue. Type B blood contains a galactose terminal residue. Type O blood is missing a terminal residue. And type AB blood has both A and B glycolipids that are expressed on the same cell. Oligosaccharides function as markers for cell identity and as protective barriers. This video was produced by the Biology Education Animation Research Project at Brigham Young University, Idaho. Lecture notes were provided by Dr. Stephen Christensen. Video animation, voice narration, and figures were also created by Dr. Christensen.